Well, first of all, Tanya and Chitske, thank you a lot for organizing this meeting. I realized yesterday how wonderful it is to have a meeting only on Kleefstra syndrome. Working on it already more than 12 years. It's actually the first time it really happened. So it's great. Don't need to give a big introduction on Kleefstra syndrome, and we can dive right into the science. And what I would like to do today is actually showing a little bit our approach, how we can use leverage on iPS cells, so stem cell models to try to understand a little bit more about the neurobiology of Kleefstra syndrome, but also other rare syndromes. And so in the back you see these beautiful cells we can generate. In red you see astrocytes, in white you see neurons. And basically we can generate them directly from stem cells from every individual patient. And that's, of course, the biggest advantage of using iPS cells is that now we don't have to generate a knockout or doing genetic manipulations. We can just take blood cells or fibroblasts from patients, transform them in iPS cells, generate the cells of interest, and of course those cells of interest then have exactly the same genetic background as the patient cells. And this is really relevant because we do know that for the rare disorders, even with highly penetrant genes such as EHMT1, that there is quite a large clinical variation, and we do not always understand it. And I think Dimitris will tell us a little bit later that the mutation by itself does matter. But also the genomic background of every individual patient can play a role. So that's kind of I would say, called the opportunity of the iPS cells. But while being working on iPS cells now for a few years, I also realized there are quite some challenges. And one of the challenges is ultimately that we aim at really having this disease model in the dish that is relevant for what we see in patient, because ultimately, we want to learn something that tells us something about the patient. So I will tell you a little bit about our approach to have this disease model in a dish. And one important concept that probably many of you are familiar with is this concept that in neurodevelopmental disorders, or actually many neurological disorders, there might be this imbalance between excitation and inhibition. So my lab has tried to develop methods to allow to measure this also in a dish and have this at least an outcome measure for this excitation inhibition balance. Now, beside that, I think one of the important challenges is when we differentiate the iPS cells to a certain cell of choice. Initially, we just were happy we could generate a neuron. We also now know that there are 100 different types of neurons. At the same time, for these kind of disorders like Kleefstra syndrome, we do not have a very clear, I would call it cellular loci. So we do not know per se which cell type is now leading to the cognitive deficits. It's very different than Parkinson's disease, where we know very well what is the cellular problem. So which cells should we make if we can make every cell we want? So that's actually really a challenge. And so what really can help us is look, for example, at a gene expression. So where is the gene expressed? Which cell types? And now we live in an era of omics technology, and so we can have access to this kind of data. And what you see here is an analysis that was done by a colleague of mine, Associate Professor Lotte Witt at the Radboud, and she looked just at postmortem single cell gene expression data, and we looked at all the genes here, intellectual autism genes, as called by the Safari project, but you know that those are mostly neurodevelopmental disorders genes. And I think what you can beautifully see here, that cell type matters. You see that different classes of neurons, inhibitory, excitatory neurons on the top, and in red you see when the gene is more highly expressed, and then you see that there is a group of genes that is more highly expressed in neurons, for example, but you also have a class of genes that is, for example, highly expressed in astrocytes, like those genes here, but then you also have genes that are expressed in microglia. So this can give us a hint to which cell type we should be looking at. And that's also a little bit the approach that we are trying to take when we try to model these kind of disorders. Now, the way we do this is, there are different ways to do this. One of our preferred way is really to differentiate then iPS cells to those different lineages. So we generate glutamatergic, GABAergic, serotonergic, dopaminergic astrocytes, also now with microglia, with Professor Lotte de Wit. And so we're trying to then look specifically at those cell types. And so one of the first cell types which I think has been adopted very a lot by the field, is a generation of excitatory glutamatergic neurons. This we can do it in a quite efficient way. It takes relatively a little amount of time, and it's very robust in our hands. And I just want to share. I mean, we are very used to do this in the lab. Every student that comes, they learn how to differentiate the cells. Starting from iPS cells to neurons. And I always think it's quite amazing that we can do this. And I just want to show you a movie. And what you will see is just cells when we plate them, iPS cells, and then we'll just image them for the first 5 days. And you'll see how quickly actually those neurons can turn into neurons. So you see on the bottom there, you can see the time. And so you see how quickly actually cells after 1 day already get these protrusions, dendrites, axons, and even already after 5 days they form these beautiful networks of neurons that connect to each other. Now, this is a very fast process. Of course, it's not a natural process, but it allows us at least to have neurons in a dish. Those neurons form beautiful synapses also. This takes a bit longer, about 20 days. And of course allows us to look then whether there are any changes depending on the genetic background. Now, this kind of approaches is looking at the morphology, but does not tell us so much about the functionality. And I think that therefore Hans has nicely introduced already the approach that we take in the lab to then do this differentiation on those what we call smart dishes, where we can actually measure non-invasively record the activity that these neurons exhibit and during development. And so one of the characteristics is really that upon development, neurons will start to make synapses and then they will start to fire together. And this firing together you really can see as a network activity and is really a proxy for synaptic maturation. You can see here on the right this image that Hans showed yesterday, where all the neurons firing at the same time. And then we can transform that in what we call the raster plot on the bottom. And there you nicely see this stereotypic development where on let's say 2 weeks, you see those black vertical lines. That means that neurons do fire action potentials. But it's only after 3 weeks that they start to do that really in a coordinated fashion. That's what we call this network activity. And this network activity is of course something you can say, well, maybe that's something very artificial, in vitro, and I can completely agree with you in one sense. However, we also know that this happens during brain development. Actually, when the brain develops, there is a lot of coordinated activity. And that's actually very well documented in the preterm babies because it's really a characteristic of the developing brain. And what you see here are actually also network activity, synchronized network activity across different brain regions. And so the idea is that this is kind of a way for the brain to develop, to really generate enough activity. Once that you have to start learning, let's say, then you get the desynchronization. So you lose this network activity because there is in principle not much information to gain out of network activity. So this is the methods that we use. But as said, we are actually really interested in developing methods to look at this excitation-inhibition balance. And so over the past years, we have developed methods then to also generate these different types of neurons. And the idea was really to have this very versatile model system that you see on the right top, where we can actually play around with the cells from patients or healthy controls, make excitatory neurons, inhibitory neurons, and then mix and match them in any kind of combination that you would like, with the idea to understand what is now the contribution of every cell type to a network deficit. And so for this, we had to generate those different cell types. So this is published. I will not go into details, but the bottom line is we can generate those GABAergic neurons, which is not per se a very easy feat to do. But they look beautiful. As you can see here in green, those are the GABAergic neurons, and in the red cells that are not green are the excitatory neurons. And the nice thing with in the method that we use is that you can actually generate them in a way that we want. So we can also play around with the ratios. We can play more or less of each cell type, and then do this on those dishes. Now, for the people that are really interested in the GABAergic neurons, we generate different types of neurons, and I'm happy to talk about that after my talk if you're interested. But how do we do this? So we have now excitatory neurons, inhibitory neurons. We can mix them together at the ratio that we want, and then we grow them on this network activity. So one of the first questions that we had is, if we want to measure excitation inhibition, can we now extract certain network features that actually can tell us something about this balance? And so one of, I think, very elegant experiment that we did was actually to vary the amount of GABAergic neurons. So on the left, you have a culture with only excitatory neurons. So you see this nice network burst on top there. And then the more you go to the right, we increase the amount of inhibitory cells. And maybe you don't see directly some difference between those Western plots, but if you now look at the bottom, I hope you can see it, we zoom in on certain characteristics of what we call the network burst length. And then I think you can nicely see that the length of the network burst actually decreases as more inhibition that you add. And if you now would plot this, so you plot the length of the network burst in compared to the amount of GABAergic neurons you have in your dish, you have this beautiful correlation. So you can see the more GABAergic neurons you have in your dish, the shorter your network burst becomes. And that's also not so surprising. This has been shown also in rodent cultures and so on. But this, being able to recapitulate this in this human system now really also allow us to look at this particular parameter to investigate excitation inhibition balance. Now you wonder, when is he going to now talk about Kleefstra syndrome? I'm getting there. So we have this method, and so we have cells of Kleefstra patients, of course. And so we looked at those 4 different conditions. Yesterday, Hans already talked about the excitatory neurons only, but here we have those 4 different conditions. And on top, you have a wild type, so healthy condition. And on the bottom, you have actually the condition where you have Kleefstra patient cells, excitatory and inhibitory. And bottom line is that you can see that if we add in the second line inhibitory neurons from Kleefstra patients, there's not so much happening. There is a slight increase in network activity, suggesting that there is less inhibition. On the third line, we have the condition where the excitatory neurons are from Kleefstra patients. And then you see this phenotype that was described yesterday. We see this very long network bursts. But now if you look at the last line where we have both conditions, then you see really an exaggerated phenotype. And that really indicates that the way how we interpret is that there is a function for EHMT1 in both cell types, the excitatory and inhibitory cells. And now you can analyze many properties of those network activities, and you can do a type of principal component analysis, which you can see on the left here. And this is actually nicely shows 4 different groups. So that means that indeed both cell types contribute to the network phenotype. And if you look at this graph, focusing on the network burst duration, which is a measure for this excitation inhibition balance, you see very clearly in the orange bar that there is this increased length, indicating that there is actually a lack of inhibition in these cultures. And we can also measure this. And indeed, if you start measuring the strength of the connections between the different electrodes, which represents actually a proxy for cells, you see that we have less inhibition in cultures that have Kleefstra syndrome. So this was, and so this would balance then the AEI ratio to more excitation. Now, this is nice, but what's the mechanism behind this? So one of the important developmental aspects of this, of GABAergic signaling, is that GABAergic signaling initially is actually excitatory. And this comes because there is a high chloride concentration in the cell. which you see on the top left. And this high chloride concentration is actually caused by the expression of two transporters, the NKCC1 and KCC2, and they actually, during development, they will change expression. KCC2 will really increase in expression, and so there will be more chloride extrusion out of the cell. And when there is, which leads to lower chloride concentration. And so when there is lower chloride concentration in the cells, then GABA only then becomes inhibitory. And so we can measure that in this cell system. And so if you look over time at this ratio of KCC2 and KCC1, you see beautifully that there is this shift occurring between day 42 and day 49. And what is nice is that you can actually also correlate that to function. Now, we can use this shift to also compare what happens in control versus Kleefstra syndrome. And so what we notice is that in Kleefstra syndrome conditions, the orange bars, the shift does not happen, or at least it does not happen as fast as in the control conditions. So that means that in normal development, when GABA should become inhibitory, at least in our in vitro system, we think that this shift is delayed. And this is not only limited to Kleefstra syndrome. This actually has been shown also for Fragile X, for example, but other neurodevelopmental disorders. And although in our in vitro system, this shift might be shifted with quite some days, in vivo, the fact that the shift might be delayed just by a few hours or one day can have dramatic consequences on how the brain is actually wired. And so having now the possibility to measure this in vitro and potentially also try to counteract this phenomenon gives us, I think, great opportunities. At the end, of course, we have to realize that we are just looking at an in vitro system. And so therefore, when I go back to my very basic scheme, one of the important things we also try to do is try to correlate whatever we find in the dish also, if we can, to in vivo situations. And so, of course, we heard about the fly models yesterday, but we also have the mouse model for EHMT1. And so the data that indicates that there is something going on with the development of GABAergic neurons also led us to investigate that in the mouse model for Kleefstra syndrome, which is a haploinsufficiency model for EHMT1. So I'll just show you one piece of data where we actually imaged a whole brain of wild type or EHMT1 heterozygous mice, and then we looked at the different classes of GABAergic neurons. And then we imaged the whole brain using brain clearing and light sheet microscopy. And you see here a very basic pipeline. The paper just came out if you are interested in. The difficulty is really to map all the cells that we find onto the brain and to quantify it. And so for that, we generated a very nice pipeline. But more importantly, if you start doing this now for Kleefstra, the mouse model for Kleefstra syndrome, we see actually really dramatic differences in the development of the GABAergic cell types. We only looked at three most prominent GABAergic cell types, the parvalbumin, the somatostatin, and the VIP. And what you see here are actually, when it's red means that there are more cells than compared to the controls, blue, less cells. And you see that already depending on the cell type, but also the brain regions, we see quite some difference between wild type and Kleefstra syndrome lines. So this really, I think, shows that the complexity of everything, because previously we would look, for example, only at one little part of the cortex and then make all kinds of assumptions, but we see that it's much more complex, that different brain regions seem to be affected. But again, clearly a change in GABAergic signaling. Now, if I go back to my gene expression data and zoom in on EHMT1, then, well, if you can see this, you can also see that EHMT1 is expressed indeed in excitatory inhibitory neurons, but actually more expressed in the astrocytes. And so that actually obliges us also to look at those cell types. And I will, for due time, not go into details, but what we are doing now is actually looking at all the different cell types that we can generate in the lab. And here is a summary of many years of work. Basically, every cell type is a full PhD. But clearly, EHMT1 is ubiquitously expressed. It's highly expressed in astrocytes, so there is definitely a role for EHMT1. And one aspect, I think, why it is important to really use the right cell types is also that if we think about therapy, it really might be important. And yesterday we heard in the introductory talk that, for example, olanzapine is one possible treatment, at least for the regression aspect of Kleefstra syndrome. And so we had the idea to do kind of a reverse experiment, in the sense like, if olanzapine has a beneficial effect on the patients, can we find this back in any of our model system? And there was also many years of work. At the end, we tested this on many cell types. And we found that at the end, only olanzapine had a very beneficial effect on astrocytes that lack EHMT1. So this work is still in progress, and I hope we'll be able to discuss that with you very soon, maybe next year. But I think it really shows how important it is also to use the right cell type. And so the astrocytes, in the way we think now, is that the astrocytes might play a role through a neuroinflammation pathway in this whole regression aspect of Kleefstra syndrome. Now, very few, 1 or 2 minutes for the last slide. We are, and I really want to show this, is actually we are now in a larger consortium within the Netherlands, which is called Brain Model, and involves also Tjitske Kleefstra and Arjan Bouwman, where we are trying to now develop those kind of brain models, in vitro brain models, for understanding disease. And so obviously it's based on iPS cells, but in the same line of my talk, we really try to understand which model complexity do we now need actually to have an in vitro model that really tells us something about the patient. And so the genes that are that we focus on are EHMT1, so that's good news, but also some other chromatin remodelers that we focus on. But most importantly, I think what makes this consortium special, and really links also with what we heard yesterday, is that we now try to link what we measure in vitro directly to what we can measure in patients. And until now, what typically happens is that we have iPS cells from a patient, where we know they have a mutation, that we have some clinical data, but we often cannot connect it directly to another brain measurement. And so here what we do, we try to really select patients with a known genetic mutation, EHMT1. We have them have an EEG, and then from the same patient, we also generate iPS cells. And the nice thing is that from both levels, from the EEG level, we can actually measure or extract EI components, so the AEI balance, but we can also do that in culture, as I showed you. And so I think this will, for the first time, be able to really correlate both our in vitro system with patient measurements, one-on-one. And if I say correlate, we don't know if it will correlate. It's a big question. But I think it is the way forward. And we have to do that not with N is 2 or 3, but we have to do that with large numbers. And so the idea is to do that with about 40 cell lines. The great thing is that we can indeed extract features of the microelectrode array that resemble actually measurements that you can also extract from the EEGs, and that's called the local field potentials. And so from both we can extract this AI balance. If you are interested, you can look at the website, and we kind of try to keep everything up to date. And one way to do this, and this is really my last slide, is that we try to also leverage on in silico models. And with that, I mean we can measure a lot of things in vitro, as I showed you, but with people in Twente, we are also able to now model what we measure in vitro also in silico. And just to show you how this looks like, on the bottom you have a network activity measured in vitro, and on the right is what we were able to generate in silico. So it looks quite similar, I think. So then the next thing, of course, if you can do this, you have all kind of parameters you can tweak in the in silico model. So what we now do is we actually mimic then our Clavestar network and try to turn on the specific parameters in our in silico model, and that actually can then predict which channel, which glutamate receptor, whatever it should be affected, and that gives us a much more targeted investigation into the cellular mechanism for each of the disease models. So this is the approach that we are taking. This is this will still take some time to further develop, but this is actually already based on excitatory-only cultures. Now, you can see there are many aspects of the research, so there's many people to thank. I have an amazing group of young, talented people. Of course, collaborators, Tjitske, Hans, Dirk Schubert in Nijmegen, and many others, and the funders. And I thank you for your attention, and I apologize for being a little bit over time.
Thanks, Neil, for this excellent talk. Are there any questions from the audience?
Thank you very much. Very nice talk. I don't know, I have a curiosity. Maybe you mentioned it, but what's your starting population to generate the neurons? Is it fibroblasts or?
Both. We have fibroblasts, blood cells. Okay. We have done an analysis on controls where we compared cells, networks that were started from fibroblasts or blood cells, and we never really saw a difference.
Okay, so which one is easier?
Well, depends who you're talking. I mean, from which perspective? Of course, from perspective from the patient, I think that blood is easier, right? But the generation of IPS is still easier from fibroblasts, pure technical, but there is little difference at this moment. So
okay, thank you.
Yeah.
Other questions?